Thank you so much for the great introduction and the great opportunity. I had so many great conversations today, and I look forward to more tomorrow. Yes, so I'll be talking to you about the work that we've been doing to understand the predictability of evolutionary trajectories using threespine stickleback. So this is a planetary model of our solar system called the Ori. It was used to simulate and predict the occurrence of solar eclipses, and researchers were able to construct this model based on a thorough understanding of the movement of planets and moons. And so this model allows us to precisely predict planetary events, such as the one depicted here. So for September 4th, 2100. In astronomy and physics, we often have models that allow us to make these types of robust predictions. However, in evolutionary biology, our models do not currently allow us to predict evolutionary outcomes with this kind of precision or accuracy. In fact, our model of the evolutionary adaptive machine is largely still a black box. But we are striving towards a level of understanding like we have in physics that would allow us to accurately and precisely predict evolutionary outcomes. And so to build our model, we really need to focus on what's inside this adaptation machine, turn it from a black box into actual machinery to understand the key factors that affect our ability to predict evolutionary outcomes and the relative effect of deterministic versus stochastic factors. And our ability to describe this adaptation model or machine is really dependent on how complex the underlying machinery is. So if we have a really simple architecture of that machine, we'll have a much greater ability to predict evolutionary outcomes. Whereas if we have a highly complex and redundant machinery, it will be much more difficult to make progress. So why do we want to be able to predict evolution? So from the academic perspective, we perhaps want to be able to test the accuracy of our scientific model and really try and validate our knowledge of the situation. However, in the real world, there's also many contexts in which there are scenarios where we might want to predict a particular evolutionary outcome. So for example, when examining patterns of adaptation in the face of climatic change or stressors, or in medical scenarios where we see antibiotic resistance. So how do we make progress? The first step is really to examine empirical patterns of evolution and ask whether evolutionary outcomes are indeed predictable. So we survey that empirical data and kind of figure out how it falls on a continuum of high repeatability to non-repeatable pattern. And there's a couple different options for achieving this. The first is using laboratory experiments. So here, something that many of you in this institute are familiar with, you have replicated selection applied to identical starting populations conducted in the lab. Often this is done with microbes, but Drosophila are also a really great model for doing this type of work. And we have the same selective agent applied to all of these replicates, and then we ask how often we see the same evolutionary outcomes. And this can be observed for phenotypes. So for example, looking at how often resistance evolves in the face of an antibiotic, which is applied to distinct evolutionary lines or replicates. Oh, I have it popped in here, our fly example. But this can also be achieved for genotypes, so looking across independent replicates and asking how often the same mutations are actually fixating in response to a common selective agent. The perhaps messier option, and perhaps more difficult, which is why I've chosen to take the path, is to let nature do the experiment. So go out, survey natural populations, and then ask how repeatable evolution is in response to similar selective conditions. So for example, you can go out to similar ecotones that exist either on closely related populations or even distinct species and ask how often the same phenotype arises. So for example, what we see here with regard to soil coloration and pigmentation in lizards and mice. Or we can survey natural populations to ask how often the same genotype underlies a repeatedly evolved phenotype, as has been done in threespine stickleback, looking at lateral plate phenotypes, which are repeatedly reduced in freshwater and have been shown to be responsible for the same allele has been found to be responsible for this phenotype. After we have kind of quantified the repeatability of phenotypes and genotypes in either laboratory or natural systems, the next step to make progress in our understanding is to identify the factors that determine the repeatability of these evolutionary outcomes. And here it's important to remember that evolution is highly complex, and this adaptive machine might in fact have many, many different parts. So understanding the genetic and ecological factors that are interacting will be very difficult, and progress might be very slow. So I'll show you nearly a decade of work in which we've made progress, but we still have a lot to do. So there's many outstanding questions with regard to evolutionary repeatability, some very basic with regard to how does evolutionary repeatability differ across biological scales from phenotype to mutation to genes, what genetic and ecological factors affect the observed degree of repeatability, but fundamentally, do patterns of repeatability allow us to predict future evolutionary responses? So today I'll focus on our work using the threespine stickleback to understand repeatability of various phenotypes and genotypes and identification of the factors that affect predictability. And the talk will be organized based on these two goals. So assessing and then trying to explain the variation that we see. So first, a bit of background on the utility of this system. So marine stickleback colonized newly formed freshwater populations at the end of the last ice age, so about 10 to 12,000 years ago. This colonization happened across tens of thousands of lakes throughout the northern hemisphere. So each of these lakes acts as an independent evolutionary experiment or replicate that allows us to assess repeatability. So in each of these new freshwater environments, stickleback have adapted to a variety of novel conditions, and there is extensive genomic resources that allow us to quantify and characterize the underlying genetic architecture. So because of these tools, we've been able to make some really great gains in the system, and there's some kind of famous or classic cases in evolutionary biology that are born out of the stickleback system. So perhaps some of you are already aware of an example where we see really highly repeatable genetic architecture underlying an adaptive trait. So an example of this is the loss of lateral plating in freshwater stickleback relative to their marine ancestors, which has been shown to almost invariably be due to fixation of a particular allele at the ectodisplacent locus. So we have this really, really repeatable phenotype and a really repeatable genetic underpinning of this phenotype. In threespine stickleback, we also see high repeatability for the loss of the pelvic girdle. So it's this structure here. I'll try and use the pointer. Here on the stickleback, in limnetic stickleback and the ancestral marine stickleback, we see a robust pelvic girdle and pelvic spine. But in multiple different benthic populations, we see the loss of this structure. And when people have surveyed these independent benthic populations, they find that the PIDX1 gene repeatedly explains the loss of this phenotype. So we have these instances of really high repeatability and perhaps predictability of evolutionary outcomes. But what about traits with more complex genetic architecture? These are single morphological traits where we have a single locus that explains a tremendous amount of the variation. What's happening when we have something that's much more polygenic? So I had a student come into my lab that was really interested in looking at climatic adaptation. And so we've been using this as kind of a complex phenotype to try and look at the genetic architecture of adaptation and how repeatable it is across broad landscape scale. So what we have done here is we've surveyed variation across gradients, climate gradients in 2 different regions, and we're asking how repeatable the underlying pattern is. So to make progress here, we've sampled stickleback across multiple climatic gradients. We've aggregated climatic variables that we've gleaned from satellite data in the World Climate and TerraClime databases. And we've reduced the dimensionality of that data using PCA because many of these variables are highly correlated. We've then prepared whole genome data, so single individual whole genome resequencing data for all of our individuals, and then we put them through our standard reference-based bioinformatic pipeline to call single nucleotide variants. We've then undertaken an environmental genome-wide association approach using a program called BayPass, which perhaps many of you are familiar with, but essentially it's a linear regression-based approach which looks for an association between a variable, such as temperature here, and the allele frequencies across your replicate populations. So if you had a locus in the gene, and it repeats this for every locus in the genome, while taking population structure into account. And so a locus that would have a positive association between a variant would look like this, where you have populations that have fixed a particular allele in warm climates and a set of populations that have fixed the alternative allele in a lower climate. And then since you're running this across the genome, you can summarize this genome-wide, where you have the significance of the association on the y-axis and all of the regions of the genome on the x-axis. Once you have those candidate genes, you can treat them like any type of candidate that you do, characterizing the molecular functions, looking at enrichment on particular regions of the genome, etc. So this is what our sampling looked like. So for a variety of different projects, my lab has been working to aggregate a really comprehensive survey of stickleback populations along the west coast of North America. The southern extent of the stickleback species is Baja California, so just south of San Diego, and they go up all the way to Alaska. So far, we've sequenced 140 populations of stickleback from just north of Baja to northern British Columbia, Canada, and we're waiting to get samples this summer that extend through out Alaska so that we have a really comprehensive survey of the entire West Coast. But for the purpose of looking at repeatability of climatic adaptation, we're going to focus in on two geographic regions. So the first is here, the Haida Gwaii Archipelago, which is off the coast of northern British Columbia, Canada. And then we have populations from California. And the reason for focusing on these two regions of the West Coast are twofold. So the first is that we have population genetic evidence to suggest that these populations have a distinct genetic origin. So populations from California share very little ancestry with those from northern British Columbia. So these provide independent evolutionary replicants in order to test for an association between climate and genetic architecture. Secondly, they actually have very similar climate gradients between the regions. So here we have a map of Haida Gwaii, where we have the geography on the left, the temperature and precipitation. And Haida Gwaii is an island archipelago with a mountain chain running down the middle of it. And this means that it has something that many islands have, where it has one side of the island that is cooler and wetter, and another side of the island which is warmer and drier. So we have this gradient, this east to west gradient. And indeed, when we take the climatic data from all of these populations that we have samples from, so each of these dots is a population that we have whole genome resequencing data from, we find that the populations on the northeastern side of the island cluster together with a warmer and drier climatic variables, and the populations on the west coast of the island have the opposite. So we have this PC1 really capturing a lot of the variance. And in fact, it's actually 60-ish% of the variation in climate is described in this west-east gradient. In California, we see a really similar gradient, but in this case, it's north to south. So in the southern part of California, it's much drier and warmer, and in the northern part, much wetter and cooler. And when we put the PCA data in for the sites that we have whole genome resequencing data for, again, we find separation along the first principal axis, and it's explaining a tremendous amount of the variation, about 50%. So really capturing this north-south latitudinal gradient, which largely is covariant with precipitation and temperature. Okay, so what we did for this environmental genome-wide association, once we had run it, we took the Bayes factor values and we averaged them across 10 kilobases, all of the SNPs within 10 kilobase windows. and then we considered windows that had a mean Bayes factor and correlation coefficient in the 95th percentile to label them as outliers. And this is what these data look like. This is just kind of the high level summary, but there's a couple take homes I want you to take home from this initial slide. So we have California on the top here, we have Haida on the bottom, we have all of the chromosomes of the stickleback genome, and then we have the strength of the association between the principal component 1 for each of these regions. And so the dots that are in orange, that indicates significant association with these climatic variables. The dots that are in black are things that are significantly associated both in Haida Gwaii and in California. So those are parallel or shared outliers or candidate regions between the two of them. So the first is that we don't have this one clean peak that's associated with climate. So this is a very complex genetic architecture. It's very similar to things that I saw in my chats today with people studying temperature adaptation in Drosophila. So it's much of the genome is responding to this shift in climate. It's not the case that there's a single chromosome or a single region. It's many, many regions of the genome that underlie this signature of adaptation. So what I'm showing here is just that we looked at the number of candidate regions, and then we did an enrichment test to look at which chromosomes they were falling on. So the data here is for Haida Gwaii. So you can see that there are candidates on every single chromosome in the genome, but there's certain chromosomes that actually have many more candidates than others. And when we test for enrichment, there was more candidate windows than expected by chance for how many windows there were on significant or specific chromosomes. So some of these were not super surprising. So 4 and 7 and 20 are kind of key chromosomes in the stickleback genome. If you read a stickleback adaptation paper, probably the gene that they were talking about or the phenotype they were talking about mapped to one of those chromosomes, because many of the ecologically important traits are also mapping to those chromosomes. We have done previous work showing enrichment of ecologically important traits with regard to diet, foraging, body shape, etc., also mapped to those chromosomes. So those weren't super surprising. But chromosome 17 was actually surprising. This is not one of the typical stickleback chromosomes. So that might be kind of interesting to look at, since that's not where we see a lot of these ecologically traits typically mapping to. When we look at California, we find, again, there's a couple of chromosomes that are enriched for candidates for climate adaptation, including 4, again, that important ecological chromosome, and 17, which is typically not one of the key chromosomes for ecological adaptation. So the takeaway here is just really that we see highly polygenic genome-wide architecture. But what about the overlap? How repeatable is this phenotype or complex phenotype? So what I'm plotting here is the number of windows on the y-axis and then the chromosomes on the x-axis. But here it's the frequency of windows that are shared between Haida Gwaii and California. And so we actually found 60 candidates within the genome were shared between Haida Gwaii and California, and this was significantly more than expected by chance based on permutation testing. So while much of the signature is unique to either California or Haida Gwaii, there are regions of the genome that are shared and associated with climate in both of the regions. And in total, it was 116 genes that corresponded to these windows. And we started digging into these candidates, and it's still early days looking at them, but there were some exciting candidates. So for example, this one gene, EIF4ENIF1, fun to say, is a eukaryotic translation initiation factor. So it doesn't sound particularly exciting off the get-go. However, it's been shown to be involved in thermal stress markers in a variety of different taxa, particularly in fish. So in salmonids, it's been shown to be differentially expressed. It is also one of the genes that has the strongest signature of positive selection in Antarctic ice fishes. So again, where there's adaptation to kind of an extreme thermal situation. And these taxa in which there's pretty strong evidence of a signature of adaptation associated with some aspect of thermal stress. There's a huge amount of taxonomic scale of divergence between these taxa. So there's 200 million years between salmonids and stickleback and 70 million years between ice fishes. So even at these broad taxonomic scales, we're finding some evidence perhaps of repeatability of thermal or climatic adaptation. Okay, so to summarize kind of where we're at with this climate stuff, we have a lot of work planned kind of as a follow-up, including looking at other climatic gradients, including data from Europe, from the east coast of North America, and from Asia. But we're already finding a lot of evidence that there is this highly polygenic architecture. I don't think that's going to differ between any of the climates that we look at. But most of the genetic architecture comparison is unique. So we're not going to be able to predict, a genome-wide response, but there is evidence of statistically significant repeatability between these independent surveys, and there are interesting candidates that are emerging that may be shared across really broad taxonomic scales. So this suggests we may be able to predictively model aspects of genetic architecture, particularly if we start digging in with regard to the effect sizes of some of these loci. And that we might be able to understand something that's biologically relevant with regard to climate adaptation. Okay, so thinking more generally about repeatability and kind of a continuum of repeatability, stickleback is a really great system because we have many different comparisons that we can make among these naturally occurring populations. So we wanted to use the stickleback system to investigate kind of the scaling of repeatability from phenotype to genotype. So to do this, we surveyed these different replicate stickleback pairs, and particularly we focus on ecotype pairs. So we have this marine ancestor, and then we have all of these derived freshwater forms. And conveniently, they tend to kind of fall along a single ecological continuum. So we call this kind of the benthic to limnetic axis, and we have these different forms that fall along it. So there are, in British Columbia, Canada, these two different ecotype pairs that co-occur in a handful of lakes in symmetry. So there's the limnetic, which forages out in the open water, and the benthic ecotype that forages in the nearshore environment. We also have solitary lake populations that kind of fall more towards the benthic. We have intermediate solitary lakes, small solitary lakes that tend to be more benthic, and then stream ecotypes, which also tend to exhibit a benthic phenotype. So we have independent replicates of these benthic limnetic sympatric pairs and also of these stream lake pairs. So what we did was we surveyed 3 benthic limnetic pairs. That's all that's currently in existence because people like to introduce things in North America into lakes. And one of the extant benthic limnetic pairs went extinct because people put catfish in it, and the other one, someone introduced crayfish, and it disrupted their mating and caused a hybrid swarm. So we only have 3 intact pairs, unfortunately. There used to be 5. And then we surveyed 4 stream lake pairs. And we stained specimens with Alzarian red to highlight bony elements, and we phenotyped 12 ecologically relevant traits. We also collected genotyping by sequencing data for 20 individuals per ecotype, and we estimated the divergence for upwards of 35,000 SNPs. Then to look at the scaling of this repeatability, we used a method called multidimensional vector analysis, which basically looks at pairwise differentiation between pairs of populations. So it looks something like this. Oops. So you have multi-dimensional data here. So you have either a phenotypic or a genotypic axis. Then you have a population of a particular ecotype that you're comparing against a population of another ecotype. And you generally have these paired by some metric, usually watershed in our case. So then there's a couple of metrics you're interested in. How divergent are these two populations that differ in their ecotype? So the length of the vector that you're drawing in multidimensional space, and what's the angle between these independent vectors to ask how similar these trajectories are between the populations. And if you have an angle that's exactly 90 degrees, that's kind of the null expectation, and that would be that there's no parallelism, there's no repeatability or similarity of significant similarity of their evolutionary trajectories. If it's above 90 degrees, it would be anti-parallelism, so they're actually doing opposite things, the two sets of pairs, and less than 90 degrees is evidence of parallelism, so similar evolutionary trajectories. So we conducted this analysis, and the first data I'm showing you here is phenotypic data. So we have the benthic limnetic pairs here, we have the stream-like pairs here, and the y-axis is this parallelism angle estimate. So less than 90 degrees, which is indicated by this broken line, would indicate significant parallelism. And what we can see here is for the phenotypes, benthics and limnetics are highly parallel. Their estimate is just upwards of 50 degrees, so much less than 90, whereas we see a lot of parallelism in the stream-lake comparison as well, but less than that, or a little bit less parallel than the benthic populations that we compare. So strong phenotypic repeatability for both of these two ecotypes. Then, interestingly, we also decided to compare the stream-like pairs to the benthic limnetic pairs because if you remember that continuum, they kind of fell towards the same ends. And again, we find evidence of significant phenotypic parallelism. So the stream populations that ecologically are similar to the benthic populations indeed have more similar phenotypes relative to the limnetic and the solitary lake populations. And in a moment, you'll see why we wanted to make that comparison. So when we look at the genomic pattern, we find a lot of parallelism. Oops, oh gosh. A lot of parallelism in the benthic limnetic comparisons here. So a degree that's about 52 degrees, so very parallel. In stream-like, we find much less evidence of parallelism, but it still is significantly parallel, so about 81 degrees. But when we compare the stream-like and the benthic limnetic pairs to one another, we find no evidence of parallelism. So this suggests that they're achieving those parallel phenotypes through very distinct genetic mechanisms. And we have done further work to show that it's the case that the benthic limnetic, the loci that underlie benthic limnetic adaptation are almost exclusively not the same ones that are underlying stream and lake differentiation, even though they're achieving similar phenotypes. So there's a lot of redundancy in the ability to generate these phenotypes. Okay, so this plot is just showing the average estimate of parallelism for genomic data versus phenotypic data, and just to kind of reinforce the fact that we see a lot more parallelism or repeatability when it comes to the phenotypic data compared to the genomic data. And this is with the exception of the benthic limnetic pairs. So they're the only ones where we see a tremendous amount of genetic parallelism. So they're kind of the outlier, and that's something that we're continuing to explore with new whole genome resequencing data. And this is just a plot showing the correlation for any given pair of populations. What is the relationship between the phenotypic parallelism and the genotypic parallelism? And we don't find a significant relationship. And what this means is that there's little predictability of repeatability from the phenotype to the genotype. So this section of data, what I really want to emphasize is just that phenotypic divergence along the benthic limnetic axis does actually seem highly repeatable, but the genetic architecture underlying this phenotypic adaptation is highly variable across comparisons and context dependent. So in the benthics and limnetics, there actually is quite a lot of predictability of the underlying genetic architecture, but for the stream-like pairs or the stream-like to benthic limnetic comparison, we don't have that same relationship. So this just suggests that predicting genetic responses across ecotypes will be much more difficult than phenotypes, which I'm sure isn't shocking to anyone. Things get a little bit more complicated once we dig into the genes and eventually the underlying mutations. So looking forward, we recently, last year, or the year before, got a grant to continue this work. And so we've been collecting a lot of whole genome resequencing data and long read sequence data to assess repeatability from the level of mutations and haplotypes to gene reuse. We're integrating gene expression data, and we're really starting to look at the source and type of variation. So we're looking at not just single nucleotide polymorphisms, but also structural variation as well. So we're pretty excited about that, so stay tuned. We're also striving toward work estimating the repeatability of natural selection across replicate ecotones and how that relates to phenotypic and genotypic repeatability. Okay, so hopefully you're already starting to appreciate the fact that in stickleback, and not just stickleback, we have a continuum of repeatability. So we see in some cases there's a high degree of repeatability, and in some cases a very low degree of repeatability. So what explains this variation? Why do we see high sometimes and low sometimes? So one thing I just wanted to assure you of is that we really do have evidence that this repeatability is driven by adaptation or by natural selection. So in stickback, when we look across different replicate pairs, we see this variation where each of these is a pair of populations. We have some cases where there's not a strong correlation between the FST estimates between divergent populations, and in some cases there's very strong correlation, so a high degree of repeatability. This is the case when we have parallel selection between the pairs that we're comparing. So if we're comparing a benthic and limnetic to a benthic limnetic, then we can find a strong degree of association in a genome-wide pattern of differentiation. However, when we have neutral divergence, so if we compare four different benthic populations to one another, so they're all adapting to the same ecological conditions, then we don't find evidence of this correlation. So we find a very low level of correlation under a condition of neutral divergence. So what we are trying to explain here is when we do have parallel selection, then why do we see this variation among populations? And one other pattern that we've observed in threespine stickleback is the fact that when we look across the genome, we find some regions that have a high degree of parallelism and some regions that have a low degree of repeatability. And so why is it the case that certain genes or regions of the genome are much more predictable than others? So I just want to emphasize that this is not specific to stickleback. So people have really similar data to what I just showed, plots like this, where we have some regions of the genome that are highly repeatable and some that are not very repeatable. So there's nice data in Litorina, in stick insects, in lizards. There's a really nice paper that I talked to some people about earlier today. Here, a meta-analysis of 17 different plant taxa. So many types of organisms, we see this continuum of repeatability. So there is a reason to try and understand why we see predictable outcomes versus not. Okay, so early in the talk, I set up this adaptation machine and trying to understand some of these cogs in an attempt to understand why we see repeatability in some cases and not in others. So one cog that we've been trying to make progress on understanding is pleiotropy, something I've talked to many of you about today. But there's really nice theory in this I can't recall the word I'm trying. There's really nice theory with regard to this factor to link it to why you would find an association between predictability or repeatability and pleiotropy. So work by Fisher suggested that there would be a relationship between the probability with which a mutation is favorable and its mutational size, whereby things that had a large mutational size down here would have a much smaller chance of being favorable. And what he was really getting at with this idea of mutational size was the number of characters that would be affected by the mutation. And this more commonly is called pleiotropy, when a single gene can influence, or mutation can influence a variety of traits. So if we extend this idea to repeatability, if the probability that a particular gene or mutation is favorable is dependent on the number of characters that it would affect, we would predict that genes with lower levels of pleiotropy would have a reduced amount of constraint and would more often perhaps be repeatable just because they'd be more likely to be fixed. So during my postdoc, I did some work to try and come up with some proxies for pleiotropy. Aside from things like yeast or E. coli, or perhaps Drosophila, it's really difficult to estimate pleiotropy directly because you'd have to go through the genome, knock out every gene once at a time, and then estimate or evaluate every phenotype that was being affected. Not super feasible in stickleback, especially with a 1-year generation time. So what we did instead was we used two proxies. One was gene connectivity, so using transcriptomic data to look at how connected genes were in a network, with the assumption that if a gene was highly connected with a variety of different other genes, it would have a higher level of pleiotropy than a gene that was less connected. We also used QTL mapping data in stickleback to look at co-mapping of individual traits, and then we estimated how many traits co-mapped to an individual gene. And in stickleback, since people have been mapping traits for almost 20 years, there's a really comprehensive database that we were able to exploit to look at the architecture. So when we use these metrics, we were able to look at the distribution of these pleiotropy proxies. So here we have the number of QTL that mapped to a particular region and the frequency. So we can see that it's the case that most genes in the genome only affect one trait, and many of them only influence a few. And very rarely is it the case that a gene influences a dozen or more traits. And when we look at connectivity, we see this kind of exponential decline where most genes are not very highly connected within the network, and a few have a high degree of connectivity. Okay, so once we had looked at connectivity and kind of described that for all of the genes in the genome, what we wanted to do is to see whether or not that was associated with whether or not a gene was a repeatably differentiated outlier between our different ecological contrasts. So what we have here is for these benthic and limnetic comparisons, we have whether or not the region of the genome was non-repeatable. So it was an outlier, but it was unique to a particular contrast, or if it was repeatable, so that it was found in two or three out of the three benthic limnetic pairs. And then we have how many QTL mapped to that region. And I'll step back. So the prediction from Fisher's theory would suggest that the regions that are repeatable would have lower levels of pleiotropy than the non-repeatable. And in fact, we found the opposite. So the regions of the genome that were more often fixed repeatedly in the benthic and limnetic pairs for alternative alleles, they tended to influence a greater number of traits than the non-repeatable regions. And when we looked at the stream-lake pairs, we found the same thing. So on average, the regions that were exhibiting some degree of repeatability influenced a greater number of traits than the non-repeatable. And when we used gene connectivity, we found the same pattern. So the highly repeatable regions of the genome tended to be more connected than the non-repeatable regions for both the benthics, limnetics, and the stream-lake pairs. But what was kind of interesting was that the most highly connected genes typically were not those that were evolving in a parallel fashion. So we are thinking that there's perhaps some sort of intermediate level of pleiotropy that's advantageous, and that's why we see it repeatedly associated with repeatable evolution. But at some point, that pleiotropy could actually become constraining. So to summarize that, we find that repeatable regions tend to harbor genes influencing more traits, and that extremely pleiotropic loci are rare in repeatable regions and may be a sign of constraint. And this suggests the degree of pleiotropy may affect the frequency of gene reuse and thus the predictability of evolutionary responses. And synergistic pleiotropy may facilitate rapid adaptation. And more recent work, including that one that surveyed the 17 different plant taxa, has is supporting that idea. So they found a very similar pattern with regard to metrics of pleiotropy and the degree to which you find repeatability, even at really broad taxonomic scales, so across these distantly related plants. So all of this evidence is beginning to suggest that high levels of pleiotropy may be constraining due to increased likelihood of antagonistic effects. And one thing we're really interested and we were unable to do in this early work is differentiate between synergistic pleiotropy and antagonistic pleiotropy. So asking whether or not a particular gene is acting on the correlated traits in a way that would be useful. So for example, if you have a mutation that confers longer dorsal spines and longer pelvic spines, then that could be advantageous. But if you have a mutation or a gene allele that's conferring longer dorsal spines but shorter pelvic spines, then that could actually be detrimental. So we're interested in looking at the directionality of these effects and seeing if that explains why we find kind of counterintuitive pattern to what theory would predict. So looking forward to the horizon, we have a lot of exciting things. Again, using this whole genome data set that we've been generating. One thing that I'm really interested in doing is testing the effect of epistasis and standing genetic variation on patterns of gene reuse. So we're trying to do some experiments in stickleback. They're hard because the stickleback lives so long, but we are generating some experimental lines where we're breeding variants onto shared genetic backgrounds to look at the phenotypic effects and trying to understand whether or not that can explain some of the empirical patterns that we see in natural populations. The epistasis becomes really quickly intangible to track when you have more than a handful of loci that you're interested in. So we're keen to talk to people that are developing new methods and theory to try and understand some of those patterns. But we think that structural variation is another really exciting place to make some progress in these questions too. There's new and exciting methods with regard to pangenomes to try and look at structural variation with regard to copy number variation and how that influences repeatability and kind of overall patterns of genome-wide differentiation. And we've collected some cool gene expression data that hopefully we'll have out soon. So overall, we're generating the data necessary to test our ability to predict evolutionary trajectories in nature. We're making progress in understanding some of these cogs in the adaptation machine to hopefully make progress, but it is slow and there's lots left to do. So I'm very heartened to hear that many of you here at this institute are interested in these types of questions and making progress using Drosophila, which is a very powerful model for these questions. And of course, I didn't do this work all alone. So I have many people to acknowledge. So David Ferrante and Caitlin Wise are PhD students in my lab. Denise Meyer was a former undergraduate. Andreas Herrer and Zach Tobias are postdoctoral fellows, and Andreas recently left. But Zach and we have a new postdoc that's taking over the project to hopefully make some good progress. And then you don't acquire stickleback samples from, or survey stickleback from the entire west coast of North America by yourself. So I have many collaborators to thank for contributing samples in that work, and then all of my funding that has paid for all of that sequencing. And then I have to thank you for your great attention today. So thanks so much.